Today we are going to tell you the story of Ian the cell going through the cell cycle.
But before we begin, let's go over some vocab. Chromatin is DNA wrapped around histone proteins, forming nucleosomes. A chromatid is one of two identical copies of a replicated chromosome, joined at the centromere. A chromosome is two chromatids connected by a centromere. A centromere is a constricted region on a chromosome that holds sister chromatids together. Spindle fibers are a collection of microtubules and proteins from centrosomes that help the cell retain its structure during mitosis. A nuclear envelope is a double membrane surrounding the nucleus of a cell to help regulate material entering and exiting the nucleus. A cell membrane is a protective, semi-permeable membrane surrounding a cell made from lipids. A centrosome is an organelle that produces spindle fibers for the meiotic spindle. Now, some background. Ian is a diploid cell and has a diploid number of 2n equals 6, where n is the number of chromosome sets present and 2n represents the two homologous sets of chromosomes Ian has, with 3 chromosomes in each set giving us 2n equals 6. The cell cycle has 4 phases: the gap 1 phase, the synthesis phase, the gap 2 phase, and the meiotic phase. The gap 1, synthesis, and gap 2 phases are collectively known as the interphase. The meiotic phase has multiple parts to it: mitosis, which is made up of 4 stages: prophase, metaphase, anaphase, and telophase, and the second part of the M phase, cytokinesis, where the cell divides.
In G0, the cells will rest and exit the cell cycle. They will continue to perform their usual functions but will not focus on cell division. A cell can temporarily enter the G0 phase due to a lack of nutrients or signals such as a neuron, but can also sometimes re-enter if it's a signal to grow when it is needed to. The G0 phase allows cells to rest or differentiate.
Welcome to G1 101, which stands for Gap 1. Here you will grow and become ready for DNA synthesis. Here's a demonstration of this going on right now. So, first of all, what you see here is the phospholipid bilayer swelling up and expanding to make room for all of the subsequent growths that are going to occur. While that is happening, the cell is also producing extra mitochondria, ribosomes, RNA, and other necessary building blocks to make sure there is enough for both daughter cells after cytokinesis is done.
What's cytokinesis?
Patience, young Padawan, everything will come in time. Now onto the final part of the Gap 1 phase. The cell initiates a sort of a checkpoint called the restriction point, in which there are 2 main proteins involved in this: CDKs, which is also present in the cell, and the cyclins, whose level fluctuate throughout the cell cycle. During the G1 phase, specifically, cyclin levels are high, which means that they will form more of something called the cyclin CDK complex, which is the protein driving almost all growth in the G1 phase.
So all I have to do is eat food?
Yes, that is correct. Now eat more food. Hello class, today I am going to be teaching you about the S phase, aka DNA synthesis. This is where you will learn to replicate your DNA so you will be ready to divide later on. First of all, you have to duplicate your singular chromosome so it becomes 2 identical sister chromatids inside your nucleus so you can divide later on in mitosis. Second of all, you have to duplicate your centrosomes or act as bases for the spindle fibers, which we will talk about later. Look inside your DNA, Ian. Do you feel your inside churn and fill? That is what you're supposed to feel.
Hi class, this is G2, also known as Gap2. Today, you will be preparing for cell division by producing the proteins and materials needed for mitosis. G2 is very similar to G1. In G2, the cell focuses on duplicating organelles and synthesizing specific proteins to make spindle fibers which separate the chromosomes during mitosis, which Mr. Snow will get to next, and checking that DNA replication was completed correctly.
Hello, we'll be covering the great phase of mitosis today. Mitosis is the process in which the nucleus divides, producing 2 genetically identical nuclei. This phase has 4 subphases, and I will have each of my assistant teachers talk about subphases.
Good day to you, class. My name is Miss Agelia, and I will be your mentor for this subphase. We will be going over the prophase. The prophase is the phase where the cell begins to dissolve its nuclear envelope, and chromatin condenses in the chromosomes. The centrioles will also begin to separate, and the spindles will start forming. Hello, everyone. I am Miss Fovia. I am your metaphase teacher, and I will be teaching you today. During the metaphase, the chromosomes will line up across the center of the cell, and each chromosome will be connected to spindle fibers and attached to its centromere.
Hey everyone, I'm Mr. Cressy. Here in the anaphase, the sister chromatids will split into individual chromosomes and move apart. This is done to ensure that when the cell splits, each cell will have a full copy of DNA and prevent non-disjunction, where cells have incomplete copies of their DNA, which can cause genetic disorders.
Hello, my name is Mr. Messi, and I am going to explain the telophase to you. In the telophase, the chromosomes will gather at opposite sides of the cell and lose their distinct shape. Then two individual nuclear envelopes, you heard me, individual, will form around the chromosomes.
Hello Ian, please sit down. This class is the final part of mitosis in the cell cycle. During cytokinesis in the animal cells, the cell membrane will pinch inward and divide the cell into two halves by using a contractile made of actin and myosin filaments under the membrane in form of cleavage furrow, which marks where the cell will divide. This will cause the cytoplasm to be split equally among the two cells, with chromosomes, organelles, and nuclei.